In this talk, we're going to learn about the different evidence that is available to support the process of evolution in living organisms. In a biological context, evolution can be defined as the changes that occur in different populations of living organisms due to a variety of changes that happen in their genetic composition over a period of time. We have ample evidence to show that living organisms have changed or evolved with time. For example, the first living organisms on Earth were prokaryotes, and they evolved to form more complex eukaryotic cells. Additionally, the first organisms were unicellular in nature and have evolved to form multicellular organisms that are more complex. The process by which living organisms have evolved can be explained by the theory of evolution. The theory of evolution is not a hypothesis or a belief. In fact, in scientific jargon, when we use the term theory, it implies that it is a fact based on many years of experimentation by different scientists. Hence, the theory of evolution is not someone's fantasy, but a fact. We consider the theory of evolution as a fact because it is supported by the fossil record, comparative anatomy between different organisms, embryological features, as well as molecular genetic features. Let's look at each one of these. Fossils are the preserved remains of organisms that were living once upon a time. These organisms can be preserved in different locations as well as different media. For example, we have found organisms that are trapped in amber. Now, amber is formed from the resin that exudes out from a tree bark, and then it solidifies, trapping anything that was caught into it. We have also found fossils in the Siberian permafrost, as well as in dry caves. We've also found fossils in rocks and many other sources. In order to study fossils, the fossils should first be formed, then preserved over a period of time, and finally recovered by the person who is going to study them. This whole process is not easy. Some of the common steps of fossil formation involve the presence of the once upon a time living organism that dies. When this organism dies, its soft body structures will all decay, and what will remain is only the hard structures. Now, on top of these hard structures, sediments will get deposited. Over time, more and more sediments get deposited, and the pressure and temperature cause the hard structures to either persist and become fossilized, or they may get fossilized and then get destroyed but form an imprint on the rock above it. There can be many events that interfere with these processes. For example, we could have a situation where the dead organism gets eaten by another organism, and hence its remains are not even present in the environment. We also have the situation where the soft body parts decay, and hence if the entire organism has a soft body structure, it's not going to get fossilized. If the organism does have hard body parts, they can fossilize, but they need the right conditions in order to be preserved to form fossils. Hence, there is a possibility that we may not have record of many, many different organisms that did live a long time ago. Once fossils are obtained, their age can be determined by a variety of processes, and one of the most commonly used technique is studying the radioactive decay of different isotopes. Scientists are able to determine the age of a fossil by analyzing its organic matter. And when they analyze the organic matter, they are measuring the carbon-14 isotope in order to determine the age of the fossil. Carbon-14 is a radioactive isotope of carbon that is present in the atmosphere. This carbon-14 can be taken up by living organisms, and hence all living organisms have a certain amount of carbon-14 in their bodies. Carbon-14 is radioactive, and hence it can undergo beta decay to form nitrogen-14. Now, in the case of living organisms, even though this decay of carbon-14 occurs, because there is a constant intake of carbon-14, the organisms will still have some amounts of carbon-14 in their systems. However, when the organism dies, then that intake of carbon-14 no longer occurs. And in this scenario, we will see lesser and lesser carbon-14 present in the organisms the longer it has died. And this way, by measuring the carbon-14 present in fossils, scientists are able to determine the age of the fossil. Another way to determine the age of fossils is by measuring the age of the rocks in which the fossils have been found. And this is usually done by detecting the presence of uranium-238. Uranium-238 is normally found in volcanic or igneous rocks. As time progresses, it decays into lead, and thereby by analyzing the levels of uranium-238 and lead, scientists are able to decipher the age of a rock. Now, when we look at the different layers in which fossils are being found, we know that the younger fossils will be towards the top of the sediments, while the older fossils will be much deeper in the sediment. By measuring the uranium-238 levels in these different layers of sediments, we are able to determine the actual age of the fossils. Hence, this is an indirect way of determining the age of the fossils based on the rocks in which they are found. It should be noted that uranium-238 is normally not found in the actual fossil itself, and hence if we analyze the organic matter present in fossils, we cannot use uranium-238 for that process. The fossil record is a collection of organisms that were present on Earth before us. And by studying them, scientists have able to collect enough evidence to show that evolution did occur in organisms. The fossil record provides us with vital information on evolutionary transitions. For example, if we look at the fossil of an organism called Archaeopteryx that lived 165 million years ago, we find that it is an intermediate between reptiles and birds. The reason scientists came to this conclusion was because by looking at the fossil, it was seen that Archaeopteryx had feathers that are normally seen in birds but not in reptiles. However, it did have a tail that is found in reptiles but not in birds. Closer examination showed that it also had teeth that's normally seen in reptiles but not in birds. And hence, Archaeopteryx is an intermediate organism as the reptiles started evolving into birds. Along with the Archaeopteryx fossil and many other studies that scientists performed, we now know that reptiles were the ancestors of the modern-day birds. Similarly, based on the fossil record, scientists found that whales and dolphins originated from animals that actually lived on land. A fossil that is the ancestor for dolphins and whales had limbs that allowed it to live on land. However, it gave rise to another ancestor that loved being in water, and hence the limbs started adapting so that it could stay in water for longer periods. This ancestor gave rise to another ancestor whose limbs evolved more and more for an organism that spent longer times in water, and these limbs would not allow the organism to move effectively on land. And this ancestor finally gave rise to the modern day whale. Similarly, fossils have shown us that snakes have also arisen from animals that had limbs before. And thus, the fossil record is able to provide us with evolutionary transitions that have occurred among different animals in the past that have given rise to animals that are present today. This is true not only for animals, but for all other living organisms as well. In addition to the fossil record, there is also anatomical evidence that supports the theory of evolution. Homologous structures are structures that are present in related organisms that have different appearances and functions, but because they arose from a same or common ancestor, they show similar anatomical features. For example, when we look at the forelimb of different mammals, like that of a human or a cat or a bat, they all have different functions, where in a human it's used to hold or grasp things, whereas for a bat it is used for flying. However, the bones that make up that forearm show high similarity between different mammals, and hence they provide evidence that all mammals derived from a common ancestor. In contrast to homologous structures, analogous structures are those that have similar functions but different anatomical features. For example, if we look at the eye of a human and the eye of an octopus, they both have the same function, which is of sight. However, when we look at their anatomy is very different from one another in terms of their tissue organization. Analogous structures do not provide evidence of a common ancestor, and so even though they may have had a common ancestor, this cannot be supported through the presence of an analogous structure. In order to show common descent or the presence of a common ancestor, homologous structures provide better evidence. Vestigial structures also provide anatomical evidence that supports the theory of evolution. Vestigial structures have no apparent function in the organism. However, they do resemble structures that were present in the ancestor. An example of a vestigial structure is the hind limb bones that are present in whales. Now, as we know, whales don't have hind limbs. However, they arose from ancestors that did have hind limbs. And hence, they have remnants of those bones that would form the hind limbs. And once upon a time did indeed form the hind limbs in their ancestors, but do not do so anymore now in the whales. Another example of a vestigial structure is the appendix that is present in humans. Organisms that are related to one another tend to show similarities during early embryonic development. For an example, when we look at different types of vertebrates, during their very early embryonic stages, the embryos look quite similar to one another. However, as development advances, we are able to see more unique structures formed that are typical to those organisms. But the fact that right at early developmental stages they show similarities is indicative of a common ancestor that gave rise to all vertebrates. Thus, embryological studies also provide support to the theory of evolution. The cells of all organisms have DNA and proteins. By analyzing the the sequences of DNA and proteins, we are able to gain information about evolution at a molecular level. As time progresses, mutations arise in genes, and these give rise to new alleles. These mutations in the genes can also cause mutations in the polypeptide sequence. By comparing these sequences, we are able to determine how related two organisms can be. For example, if we look at the amino acid sequence of hemoglobin between a human and a macaque, there is a difference of 8 amino acids. This small difference indicates that humans and macaques are more closely related to one another. On the other hand, when we look at the difference in the amino acid sequences in hemoglobin between human and lamprey, there are 125 amino acids that are different. This tells us that humans and lampreys are much more distantly related than humans and macaques. Thus, greater the difference in the amino acid sequences or DNA sequences of organisms, the more distantly they are related to one another. Evolution is usually driven by natural selection, and this process is very evident when we look at the phenomenon of convergent evolution. In convergent evolution, distantly related organisms undergo changes or evolve to have an appearance that's very similar to one another. This usually occurs when the environment they are in is similar. For example, when we look at the bodies of fish and dolphins, even though one is a mammal and the other is not, their body structure is very streamlined and similar to each other. This is because both of these organisms live in water, and living in water provides the environmental pressure that requires the body to be streamlined in order to survive in this environment. Another example of convergent evolution can be observed between placental and marsupial mammals. Marsupial mammals are normally observed in Australia, and Australia, being an island, was geographically separated from other land forms on Earth. And hence, these marsupial mammals evolved on their own, while the placental mammals evolved in places other than Australia. However, when we look at the different types of mammals, we are able to see that there are many similarities between some of these mammals. For example, we have a placental mole that's present outside of Australia. However, there is a marsupial mole that shows great similarities to the placental mole. Same thing is true when we look at the lesser anteater, which is a placental mammal that's found outside of Australia, versus a numbat, which is a marsupial that is found in Australia. And so this way, there are many other examples of organisms that are present in two totally geographically different regions and are different types of organisms, in that one is a placental mammal while the other is a marsupial mammal. However, they show strikingly similar phenotypic appearances because the environment that they were in was similar, that allowed them to evolve similar features. Thus, there is a lot of evidence to support the theory of evolution, and hence it is accepted by most scientists as well as many other people in the world. However, the theory of evolution is also controversial in some groups. So we're going to look at some of the criticism against the theory of evolution and the responses to those criticisms. One of the criticisms is that the theory of evolution is not solidly demonstrated because, after all, it is just a theory. The response to that is that in the scientific jargon, a theory is a very well-established fact. And this fact is based on numerous experiments that have been performed by different scientists. And hence, in science, a theory is not a guess or a feeling based on personal beliefs, but it is an established fact. Another criticism is that there are no fossil intermediates because no one ever saw a fin on the way to becoming a leg. Now, it is true that it is hard to see a fin become a leg because the process of evolution is slow. Now, we do have many fossil intermediates that show evolutionary transitions between different organisms. However, because of the nature of evolution, it is not possible to see dramatic changes occurring in organisms in a very short period of time. Another criticism is that evolution violates the second law of thermodynamics because it's simply not possible to have more complex organisms arise from simpler organisms. And the response to this is that the second law of thermodynamics states that any process that is spontaneous results in an increase of the entropy of the universe and not just the system. Another criticism that is made is that the organs of living creatures are so complex for a random process to have produced them. If there is a clock, then there is an evidence that there is a clockmaker. The response to this is that natural selection is the driver of evolution, but natural selection is not random. Highly complex organisms were able to evolve because there were generations of small changes that were made through the process of natural selection. The presence of certain features like vestigial organs do not support the idea of an intelligent designer, and hence it is hard to gather evidence that supports the intelligent design argument. There are many other criticisms against the theory of evolution, but unfortunately there isn't much evidence to support those criticisms. With this, we come to the end of our talk where we learned about the theory of evolution and different evidence that supports this theory. This includes fossil records, anatomical structures, molecular information, so on and so forth. We also learned about criticisms related to evolution and how scientists respond to these criticisms.